Good morning. Myself, Dr. P. Sakthiselvan from Bioengineering Department, and I am handling animal biotechnology. So, in last two sessions, we have discussed about the Unit 1. The Unit 1 comprises of introduction to animal cell culture, types of animal cell culture, culture media, and organ culture. And today we will discuss about Unit 2, that is blotting techniques and monoclonal antibodies. Monoclonal antibodies, in case of therapy, comes in say third unit, and in case of applications in diagnosis, it comes in second unit. So another important thing, what are the bacterial and viral diseases in animals, and RFLP, that is restriction fragment length morphism. That is, these are the topics which comes under unit 2. So I will give you the overall introductions about the other topics in the unit 2, and we will move on to the blotting techniques. That is one of the topic in the unit 2. Blotting techniques. What do you mean blotting? Blotting is nothing but separation of DNA or RNA or protein, which is double stranded. We have to separate it and go to the move on to the carrier, then it pass on to the gel electrophoresis. So techniques for transferring DNA, RNA and protein, which involves blotting techniques. So we will we will go to types of blotting techniques. There are three types. One is northern blotting, western blotting and southern blotting. In case of southern blotting, it is used to detect DNA, and northern blotting, it is used to detect RNA, and western blotting, it is used to detect the proteins. Okay. So blotting tests. Blotting is used, commonly used is Wortmann filter paper, that is the thickness is 3 mm, and it is mostly widely used because the reason is high quality, purity and consistency. Okay. So first, before going to the blotting techniques, we should prepare the sandwich. Sandwich in the sense, all very well know about the sandwich. Sandwich is a 1 by 1 layer. Okay. So first one you see in the PIP slide, as in the figure, see bottom one and bottom is the top and bottom is glass bottle, and above that above and below the, suppose if you take the top of the glass bottle, glass plate, or below one, which is bottom of the glass plate, which is composed of paper towels. Paper towels in the means we are folding the papers and keep with it like a towel. Okay, so that is it. In the top plate we have to keep it in the below, so we will go by bottom. Okay, bottom we have the bottle plate, then that is glass plate. Okay, so above the glass plate we have to keep the paper plates, towels, then it is plastic wrap, so to fix the papers in the glass plate, that is plastic wrap. Then gel, that is SDS phase or gel electrophoresis, that is the gel. And another membrane, that is more important thing here. The nitrocellulose membrane is commonly used for all the blotting techniques. So nitrocellulose membrane, then above Wortmann filter paper, that is thickness is 3 mm. Again paper towels and glass plate. This is comprised of sandwich. Okay, that is blotting sandwich. So which is consists of filter paper, nitrocellulose membrane, glass plate and gel. Okay. So before going to the methods or different kinds of blotting techniques, we should know the history. Here the name southern blotting or the blotting was first introduced by a scientist, Sir Edwin Southern. he is introduced the name southern blotting. Southern blotting, northern blotting or western blotting. He is developed in the year 1975. First we will see one by one, that is southern blotting, next northern blotting, then western blotting. Southern blotting is used to redact DNA. Okay, the method involves separation, transfer and hybridization. This DNA can be a single gene. Part of larger piece of DNA is a viral genome. So this is the key method is hybridization. Hybridization is nothing but multiplication or it hybrid with the another cells. We see another cells which produce antibody. Okay, so that is here. We have to separate the DNA from the cells. First we will see the process. Hybridization is a process of forming a double-stranded DNA molecule between a single-stranded targeted DNA, or that is targeted DNA in the sense patient DNA. See, we have to southern blotting in the sense we have to detect the DNA. Okay, so for that is double-stranded DNA. So one standard is normal and another one is targeted. That is, we have to combine it and we have to form a new DNA. That is the southern DNA, southern blotting. Next one is principle. The principle of southern blotting, first one, mixture of molecule is separated. That is the main advantage or main principle. Okay, whatever, suppose if any compound is present by any cells, it will easily separate the particular molecules. Mainly for southern blotting it is DNA. Okay, next one, it should be immobilized on a matrix. So immobilization will be done by gel. Then probe addition to the matrix to bind the molecules, because we have to add any labeled probes so that it can easily attach to the matrix. Next one, some of the probes which are unbound to the gel, so that probes are removed. Okay, so the place where the probe is connected corresponds to the location of immobilized target molecule. That is what the important. Okay, these are the steps in the southern blotting. First we have to digest the DNA which is obtained from the cells. Then it has to be fragmented. Okay, we have to separate the double-stranded DNA into a single-stranded. Then we'll go to the gel electrophoresis, then transfer to the membrane. The membrane which we use here is nitrocellulose membrane. Then probing. Probing is nothing but we have a single-stranded DNA. Then we have to probe with another targeted or patient DNA. Okay, that is probing. Then finally autoradiograph, which forms double-stranded DNA. Okay, this is the apparatus used for southern blotting. So this slide shows or explains in detail about the southern blotting. So from see this slide. So first we have to digest the DNA, and DNA is passed to the gel. So that is DNA is applied to gel which undergoes electrophoresis, and after the electrophoresis has been done, it will be moved on to the salt solution blot DNA. Okay, so that is here petri dish which contains salt solution. In the salt solution we have to keep the sponge, and above the sponge we have to keep the gel. In the gel we have to keep the membrane, that is nitrocellulose membrane, then paper towels. That is what we have discussed in the earlier slide. So then after particular time it will be transferred, DNA is transferred, because sponge, above the sponge gel is there, above the gel nitrocellulose membrane is there, then above the membrane we are keeping the paper towels. So because of that, whatever the DNA is present in the gel, it will be moved on to the membrane. So DNA is transferred to the filter, and filter will undergo hybridization. This is very very important phenomenon in southern blotting. Okay, so here hybridization is done with unique nucleic acid probe. Okay, see this figure. In this we have, it will be sealed in a bag. Okay, take a membrane, put it in the cover and lock it. Okay, here along with the probe. Okay, here some of the probes which are combined with the single stranded DNA which is already present in the filter. Then after some time we have to take over the filter and remove unbounded probes. That is the important thing. Then after removing the unbounded probes, then it will go to the autoradiogram. Here exposed, when you keep the filter to the X-ray film, then you will autoradiogram will show you the double stranded DNA. This is the method for detecting the DNA using Southern blotting. The application of Southern blot is, it is used for gene discovery, mapping of DNA, evaluation purpose, development studies, diagnostics, and finally forensics. In case of forensics, it is major application for identifying the DNA using Southern blotting. Okay. So coming back to Northern blotting. Northern blotting is developed by a scientist called James Alvin and George Stark at Stanford University. Okay. So in case of Southern blotting, it was introduced by Sir Edwin Southern in the year 1975. Okay. So in case of Northern blotting was developed by James Alvin and George Stark in Stanford University. So Northern blotting technique is nothing but it is used to detect RNA, that is specific RNA sequences. So steps involved in Northern blotting is, first we have to isolate the RNA, then loading of sample into the gene, then blotting on nitrocellulose membrane, everything same as Southern blotting. Here we are using RNA, there we are using DNA. Okay, so undergo labeling with the probe, then washing to remove the unbound probe, and finally it will be detected using autoradiogram. This is the figure which explain you the Northern blotting. See the figure. We have the sample. From the sample we have to isolate the RNA. When RNA is isolated, it will undergo electrophoresis. So electrophoresis, that RNA is bind to the gel. Then it will undergo the Northern blotting. So Northern blotting is nothing but transfer of RNA into the nitrocellulose membrane. Okay, so nitrocellulose membrane which is moved to the hybridization process. Here in the hybridization process, RNA containing membrane is equipped with probe, which is you can say any labeled probes, which kept it on the cover and lock it, and after sufficient time or after some time, that probes which are bind to the RNA present in the filter, and it will be visualized using X-ray film by the instrument called autoradiogram. This is the simple process which takes place in the Northern blotting for detecting the RNA from the sample. Okay, so application of Northern blotting is a standard for the direct study of gene expression from the RNA and detection of RNA transcript size, study of RNA degradation, study of RNA splicing. That is very very important in application in case of Northern blotting, because RNA splicing which can detect alternatively spliced transcripts. Next one, it studies about the RNA half cycle, then it helps to study the internal ribosomal entry site, that is IRES, internal ribosomal entry site. Okay, because it is used to remove the possibility of RNA digestion versus second cistron translation. So in northern blotting, we are confirming that from the sample you have to isolate the RNA, and RNA undergoes gel electrophoresis and it will be bound to the gel, and after bound to the gel it will undergo for northern blotting. Northern blotting is nothing but transfer of gel or transfer of RNA into the filter membrane. The filter is here, what we use is nitrocellulose membrane. Then it will be coupled with labeled probes and put it in the cover and keep it for a sufficient time, and after a particular time you will remove the membranes and keep it on the X-ray film, and you will visualize the RNA which is probed. Okay, unprobed unbounded probes are removed separately. Okay, next one, Western blotting. So Western blotting, it is used to detect proteins present in the sample. Sorry, here discovery by the scientist Dr. Douglas Lake of the University Arizona School of Medicine's Department of Microbiology and Immunology. Okay, so here the technique in which proteins are separated by gel electrophoresis, then it will move on to the membrane sheet and it will be labeled with the antibody. Okay, here this is the technique works on the principle of antigen antibody relationship. See, that is a very important, because when you take about the monoclonal antibody, so we are inserting the antigen into the mouse and we are producing the antibody against the antigen. That will be discussed in the next presentation. So here that principle is that works on the antigen antibody relationship. Okay, here the prerequisite for Western blotting is SGS phase, that is gel electrophoresis. Okay, sodium dodecyl sulfate, that is SGS. Okay, it is a technique, they are prerequisite for Western blotting. So SDS is nothing but sodium dodecyl sulfate is a detergent that can dissolve hydrophobic molecules but also has a negative charge attached to it. That is very important principle of SGS. Okay, so SGS, so sodium dodecyl sulfate, it is a detergent. It dissolves the hydrophobic molecules present in the sample, which also has a negative charge which attached to it. Okay, so there are the steps involved in Western blotting. First one, gel electrophoresis. The proteins are separated according to the size, because the protein size is very in a range. If you take any protein, they may vary from 1 kilodalton, that is kDa, to 10,000 kDa or kilodalton. Okay, so first we have to separate the proteins using gel electrophoresis based on their size. Second one, membrane transfer. So from the gel electrophoresis, the proteins are transferred to the membrane. Okay, the membrane which we used here is nitrocellulose membrane, which is commonly for both southern blotting, northern blotting and western blotting. Next one, blocking. Blocking here is done to prevent non-specific protein interactions, okay, between the membrane and the antibody protein. So a blocking is done to prevent the unbounded or non-specific protein sequences interactions between the membrane. Okay, so this is the picture representation about the western blotting. See, first the sample which is undergo for gel electrophoresis. See the slide. So it is shows SGS polyacrylamide gel electrophoresis. So the proteins which are bound to the gel and it is transferred to the membrane. The membrane which we use is here nitrocellulose membrane. After the transferring of protein into the nitrocellulose membrane, it will be labeled with specific antibody. See, some of the proteins have we will easily known by the molecular weight of the protein. So for the particular protein, we have to labeled with the antibody, that is specific antibody. Okay, so after some time we will detect the antibody, that is the reveals the protein of interest. Okay, next one, blocking in waste blotting. So waste blotting, it incubated as the generic protein such as milk proteins which binds to any remaining sticky places on the nitrocellulose. That is a drawback. Okay, so an antibody that is specific for the protein of interest, that is primary antibody, that is AB1, is added to the nitrocellulose. Okay, so blocking is done to prevent non-specific protein interactions between the membrane. Okay, so it's here we are blocking the non-specific proteins such as another layer. For example, here milk proteins which binds to any remaining sticky places. Okay, so only the band containing the protein of interest binds to the antibody, forming a layer of the antibody molecules. Okay, so that is, we have to identify which protein has the ability or which protein has the layer to form the antibody. So following several rinses for the removal of non-specifically bound antibody 1 and antibody. Okay, so the antigen complex is nitrocellulose. Here the antibody 2 which binds to another protein which are non-specific in nature, so that has to be removed. Okay, so antibody is radioactively labeled or covalently linked to the reporter enzyme. Okay, which allows to visualize AB1 AB2 complex. Okay, see, AB1 and AB2, that is antibody 1 and antibody 2 are important, so that bind to the specific protein present in it. Next one, applications. So the application of Western blotting is to identify the specific proteins based on the binding property with the antibody. Next one, it is used to identify their masses, and another application is the confirmation of HIV test is to detect HIV, anti-HIV antibody in a human serum sample. So when you have the, like somebody recently, so many case studies have been very common known to all of us regarding the HIV. Okay, so it can easily detect the anti-HIV antibody in the present in the human serum sample. Then definitive test for bovine spongiform encephalopathy, that is BSE. Okay, bovine spongiform encephalopathy, commonly referred as mad cow disease. So it is used for the analyzing the mad cow disease. Next one, some forms of Lyme diseases testing also done by the Western blotting. Then protein blotting workflow. This is, first we have to select the method and select the equipment and prepare the reagent accordingly and perform the transfer to the nitrocellulose membrane, then detect and image the protein. Okay, so this is the overall blotting workflow. Here protein gel, that is SGS page, it is commonly used. Here the dye is used Kumasi blue. And faster way to Western blot is quick Western kit is there. It is used to identify the protein easily when compared to normal Western blotting techniques. Okay, so there is what is the difference between quick Western blot and standard Western blot is explained in this slide. Here it blocks the membrane for 1 hour, and it is blocks the membrane 1 hour. Primary antibody incubation and primary antibody reduction reagent. Second antibody incubation, after washing the primary antibody, it will go for secondary antibody, which is kept for 1 hour. After 1 hour, scan blot for the assay. Similarly, for quick Western blot, there is no secondary antibody incubation. So once you detect the primary antibody detection, you will go for scan blot assay. Okay, if the process will takes within 1 hour 30 minutes, that is 90 minutes. Okay, and after that you will go for identifying the proteins. So in this presentation we understood that various types of blotting techniques, that is northern blotting, southern blotting and western blotting. Western blotting, nowadays the kit is available for easily identify the protein. Okay, that is blotting techniques. And next one, bacterial and viral diseases in the animals. So many bacteria caused, for example, staphylococcus, okay, then bacillus. These are the bacteria. Virus, so many viruses, rubella virus, measles and mumps virus. These are the virus. These are the virus which are affects the animals. And similarly fungi. Fungi also Clostridium tetanus. These are the fungi which affects the animals, which creates the diseases. And restriction fragment length morphism also used for analyzing the length of the any DNA or RNA, okay, which helps us to understand the phenomena behind the cells. Okay, thank you for understanding the presentations. Thank you.